I'm going to give us just a moment or two to let a few other people sign on. And I want to thank everybody for joining us today. Is this our second in the series? I think that's my belief. So I appreciate people coming out to our, MSU CHM webinars and joining us. And I think that I'm going to get us started. So given that we have a timeline, so I want to thank everybody for joining us today. And I want to thank Jack Lipton for coming on to talk about the work he's been doing, talk about COVID and testing. It's a huge issue for us right now. Obviously in East Lansing, we've had an outbreak, and there's an interesting situation where our percent positivity as measured by MSU and by the county is staying up, but the number of cases is going down. And that's because probably the amount of testing is people coming in for testing is also going down. So this is a really timely, interesting moment for us to be talking about this. Dr. Lipton received his undergraduate and doctoral degrees in psychology and behavioral neuroscience from UC Berkeley and the University of California, Los Angeles. He studied fetal brain development from in utero cocaine and ecstasy exposure at Rush University Medical Center in Chicago, where he held his first faculty position. While at Rush, he began working with colleagues on the origin of Parkinson's disease. And in 2004, he became director of the Division of Neuropharmacology in the Department of Psychiatry at the University of Cincinnati. And there, the NIH awarded him and his colleagues a Morris Udall Center of Excellence in Parkinson's disease research. And for those of you who don't know, those Center of Excellence grants are hard to get and really demonstrate the kind of quality and breadth of work that's happening in a center. In 2009, he brought that center to Michigan State, and the College of Human Medicine and established the Department of Translational Neuroscience, and is the founding chair of that department. And they are based at our Grand Rapids Research Center in downtown Grand Rapids, just down the hill from the Psychia Center. Under his leadership as professor and chair, the TSM, the Translational Neuroscience team has garnered over $50 million in research support and is among the top research productive units at the university and the college. And in early March, he and his team started working on a COVID-19 test that's more sensitive and flexible than tests available traditionally through physicians' offices. And I think importantly, that test, they were able to modify that work to be able to use samples other than the nasopharyngeal swab that gets such bad reviews for comfort and frankly, effectiveness in getting a good sample. And has led to the work of the saliva sampling that underlies the early detection program that we're using now. In the past few months, he's authored an op-ed on COVID-19 testing in the Wall Street Journal. He's been a COVID-19 testing authority on MSNBC, NPR, and Fox News. And today he's going to be summarizing his team's work in establishing that COVID-19 early detection program, which is currently running over 1,000 samples a day. In a lab that he and his team picked up and implemented really over about 4 weeks. I have the good fortune to sit in on those meetings, and it has been a remarkable team effort, and I'm really impressed and delighted with the work that they have done. And I am excited that Jack is going to be talking to us today. So with that, Dr. Lipton, the screen is yours.
Thank you, Dr. Souza. So let me share, make sure things are working. Always fun to pick the right screen to share. Can everyone see that? Good. All right. So today I'm going to give you my biography from March till today, and it's kind of a crazy story. So we're going to talk about COVID-19 testing. And the primary question, which we were hoping to answer, can saliva tests detect a COVID-19 outbreak sooner? So what are we going to learn today? we're going to go through the early work that we've done to operationalize a SARS-CoV-2 test. I'm going to talk a little bit about the two platforms we used and why we chose the one we did for the early detection program. We're going to talk about the different matrices that samples can be collected in and how we made our decision to move to saliva. I'll talk about the pros and cons on those. Then I'm going to talk a little bit about advocacy to allow academic research labs to support COVID-19 testing efforts, which is really where I turned my frustration into a public conversation on some of the issues that I thought were really important and my colleagues thought were important. Then I'm going to go through and go into the actual early detection program, talk about the development of the Saliva Kit, the development of the pooling process that we use, which uses about only 20% of the reagents and takes about a fifth the time to do all of the work we do when we do high throughput surveillance of saliva samples for COVID-19. And then the entire system that we've developed to collect, process, and analyze and transmit results on up to 10,000 samples per week. We're not quite there yet, but that's only because we don't have enough people to give us spit. And then I'm going to go through the remaining challenges and the things that we found beyond our successes, the things that remain as an issue, which I think all of us need to think about. So here, let's first go through an overview of the timeline, just so you can understand the scale of what we're doing. And even though Aaron mentioned a few of these, let me whip through them here. So originally in March, when everything shut down, we were already talking in the lab because we have a droplet digital PCR system and we do real-time PCR work all the time. We're a translational neuroscience department and we work on gene therapies. So there's a lot of viral quantitation that goes on here. And so we were looking at the literature and things that were coming out of Wuhan, and we said we could do that. That seemed pretty simple. And mainly that was Joe Patterson, who's one of our research assistant professors. And that's what he gets for talking smack about being able to do things. And now he is changed to this program, like many of us are. So that happened in mid-March. We made a decision to develop an assay. We developed that droplet digital PCR assay within a couple of weeks and validated it with contrived samples, so basically simulating what a COVID-19 sample would look like. We started using human samples. We developed an IRB protocol to do human subject testing to compare droplet digital PCR with standard PCR. We started doing that in early April. We continued to community testing in that program and developed a larger scale so we could get more at-risk patients in the community or subjects in the community so we could look for spread and better understand the differences between the testing modalities. We published those protocols online at protocols.io. And then in May, we started optimizing for saliva. A task force subcommittee for the reopening of campus was formed in late May. And I was asked to co-chair one of the committees, which was the testing committee. And then it has morphed into this early detection program committee. Pooling feasibility to do testing of pooled samples, which I'll go into in more detail later, began in late May. We were approved to proceed by MSU in mid-June, and we received funds to start our program, turning our localized program that we did in Grand Rapids to work on our own return into a university-wide program. We completed our pilot with the College of Human Medicine and the College of Vet Med, which provided us much spit, which we needed a lot of in order to be able to do the pooling process, validated that process. The lab move-in, which is in the bioengineering building in East Lansing, started on August 11th, and we got our first sample on September 10th. So that was a 4-week sprint from buying things and getting into that building until we were running. So we've had a lot of really long weeks, let's just say that. So let me go through some of that early work. So I'm going to go through basics, very, very simple. How does PCR work? I'm not going to give a very adequate explanation, but it'll be good enough for this. I'm going to talk about the difference between droplet digital PCR and quantitative PCR, and then nasal swabs versus saliva before we get into the operationalization. So let's look at this. Here's a brief inadequate answer on how PCR works. We collect a sample, we extract the viral genetic material, and then we reverse transcribe the RNA, which is the genetic material from the virus, into DNA, and we amplify that in a machine, which is essentially a real-time PCR machine, which is a thermal cycler, which just cycles through 2 different temperatures in order to amplify the genetic material. And as it does so, it produces a fluorescent signal after each heating and cooling cycle. And that's what each of these little triangles are on here, each cycle. And then as you can see at a certain point when we get exponential growth, you start to see if there is viral material that you're looking for present, you'll start to see this exponential phase. And so you'll start to see these curves go up. This is what we tend to see, and you'll see this a few times in the talk. We wanted to utilize a slightly different method than real-time PCR, which is what I just showed you. And we wanted to use this process we call droplet digital PCR. Well, lots of people call it that. But it's more sensitive, and it offers us some different abilities. So let's go back through what we looked at for qPCR. Think about this sample where it says PCR. So we take this material out of a vial, we amplify it, as I mentioned, and we get that green fluorescent signal here, and then we can measure that over cycles in this graph that you see here, so that fluorescence goes up. With digital droplet PCR, it's a much more sensitive process. And what we're doing, if you look at the sample, we take that sample and we divide it up into tiny little droplets. And instead of doing the PCR reaction on the whole sample, a tiny PCR reaction happens in each droplet. And if one of those droplets has the actual viral material that we're looking for, it'll show up as a little green droplet. And what we do is we do these independent quantitation of each droplet, and then those are counted. They run through a bubble counter. And so we can actually get a digital measure of how many copies of the original genetic material there were. So we could say there were 10 copies of the virus in this particular sample. So it's more quantitative, it's much more sensitive. The problem was, and we used it for a couple of months, the problem was it was slower and it's expensive to scale it up. The machinery for it doesn't really lend itself to high throughput work. So what we found out was droplet digital PCR was great. It was great for small scale implementation and for validation of the quantitative PCR that we used. And we really would love to use it, but until there's a more reliable high throughput system available, we're not quite ready to do it, unless someone wants to give us like $2 million. The qPCR is more amenable to large scale endeavors for now, and that's what we decided to use. Now let's talk about sampling. So nasopharyngeal sampling, everyone hears about this, right? They take a big stick and swab your brain, basically, is what it feels like. It's very uncomfortable. That's the official gold standard of testing. We've all seen how terrible it is. Here's a nice picture that I think is pretty terrible, and I always like to show. So here, nasopharyngeal swabs, they go way, way into the back of your nasal cavity. Someone twirls it around, scrapes in the back there, and hopefully pulls a sample. The problem with this is that it requires an expert to administer. You cannot do this on your own. It requires extensive PPE. People need to, and we know PPE has been hard to come by. There's been a lot of issues, particularly when we started doing this, sourcing sufficient PPE to change that in between patients. Swab sourcing was difficult at the time, and there's variable sample quality depending on how well you do this swab. If you don't get a good sample, you may get a false negative on the test. So we started looking at other matrices. First, we did a nasal swab. The advantages of it, and the reason we chose nasal swab is because we couldn't get anything else. So we actually couldn't even get nasal swabs. We ended up using buccal swabs. So these are the swabs that you would, rub in your cheek to get some DNA. And for genetic testing, the value of these is that there's minimal discomfort. They're easier than an NP swab for participants to experience. Less PPE is needed. The cons are there's not as much material in these. You'll get a lower signal. If you're asking someone to do it on their own, self-guided sampling can be error-prone, right? People sometimes will just twirl it around in their nostril. It doesn't really touch anything. And we found that was a problem. And there was limited data on the validity of this kind of nasal swab. For saliva, the pros are there's no discomfort unless you're uncomfortable spitting. It's easier than a nasopharyngeal swab or a nasal swab for participants. No PPE is needed. We get about 10 times the genetic material out of saliva, and we can retest that sample many times. And the way that we've done it using a compound called RNA Shield, which is a preservative from a company called Zymo, there's no cold chain needed. So we can keep that sample and it's stable at room temperature for weeks, and we've tested that. The cons with saliva are the viscosity challenges for the lab. It's really gooey, which means that pipetting it's difficult, and you don't want to make sure that you're not moving it from one place to another and taking a positive sample and drooling it across other tubes because you'll contaminate them. So that's a challenge. There's restrictions on eating and drinking. You cannot have anything to eat or drink, not even water, no smoking, no chewing gum 30 minutes prior to doing the test. And some people that have dry mouth have spit production challenges. So let's go through the progress. So we were excited, right? We have a test that works. We have a matrix that works. That's great. 2 different methods that work instead of an NP swab, 2 different PCR tests that are running for us. We have an alternative sourcing of materials. So we were trying not to utilize the materials that were in such high demand at point of care facilities or hospitals or clinical testing labs. So we're using alternate materials to address those supply chain challenges. And so we were excited, let's validate this and let's offer it up to clinical labs. And what happened? Nope, can't do that. That was disappointing. And then we were like, can we run these tests on our own outside of research projects and then, maybe kind of create our own lab and offer this up into the community and help people? No, we couldn't. Why couldn't we? We couldn't because of, let's see, where is it? It's not showing up. But the word that would appear if I was to say it is CLIA. So CLIA is the Clinical Laboratory Improvement Amendments. These are the regulations associated with clinical diagnostic laboratories. And the problem that we have with CLIA is that it's there for a reason. We have clinical diagnostic laboratories to provide us with very consistent testing. If you want to get your cholesterol checked in New Mexico, you're going to most likely get the same number if you go to New York. And if we don't have that kind of reliability, we cannot trust the tests that we run. The problem is that when it comes to SARS-CoV-2 and COVID-19 testing, we didn't have nearly the amount of capacity that we needed. And so I got really frustrated over the fact that we couldn't become a testing facility. We couldn't develop tests on the behalf of other clinical diagnostic labs because we can't even validate a test without FDA approval. And for us to get FDA approval, we have to be a CLIA lab. So it's kind of a catch-22. And so that made me a little angry, like I get, and oh, there it is, hey, CLIA. And so I decided to become an advocate for this cause. And so the first thing that I did was working with my colleague, Carol Sortwell, who's one of the PIs on our IRB for testing people with our new assay, was to write an op-ed. And so we had this coronavirus test, and we wanted to use it. So I put out this op-ed in the Wall Street Journal. The Wall Street Journal was very supportive of that because they are not necessarily fans of regulation. And so I was saying we should be easing these regulations and allow academic labs to do that kind of testing. And that led to other opportunities. By writing this, other people contacted me, and I was able to parlay that notoriety into new avenues to talk about this problem. So we talked on Fox about how academic labs can help with COVID-19 testing. We talked about the challenges of CLIA on MSNBC. And then we ended up talking more recently on the Rachel Maddow show on MSNBC about the program that we developed. And so in the process of doing this, we got the attention of the Michigan congressional delegation. And Kathy Wilbur at MSU and Government Relations was really key in this. We got together with the Michigan congressional delegation, both a bicameral and bipartisan group, to lobby for changes in the regulatory framework that would allow academic labs to do testing. And they helped us, and other people around the country were doing something similar. I know this was going on at Cornell. And then as a result of that, it paid off. And so in June, there was a testing blueprint that had come out from the White House, and then they made an addendum to that. And it was very specific to what we were trying to do. And so this blueprint was called Opening Up America Again, sponsored by the White House, signed off by the CDC and the FDA. And it laid out the specifics for academic laboratories to help in the fight against COVID-19. And I underlined just some highlights here. So college and universities can use their research testing capacity to support practicing surveillance testing on campus. They can use various platforms to run maybe 500 to 1,000 samples per day. And their student bodies and testing their student bodies on faculties on a regular basis, particularly if the incidence of disease is low enough to enable effective pooled testing. And we're like, hey, this is pretty good. So we moved forward on this surveillance. But what exactly is surveillance? And I think it's really important to understand the difference between surveillance, diagnostic testing, and screening. So let's talk about those. Clinical diagnostic testing is what we were talking about, right? A laboratory test ordered by medical personnel to provide medical information or diagnostics for an individual conducted in a CLIA accredited lab. Okay, I get that. Screening. What is screening? This is something that we deal with this line all the time, where screening ends and surveillance begins. Screening is utilizing the clinical diagnostic testing system, just like in the clinical diagnostic test above, to look at specific measures that are typically used to evaluate disease states, but not necessarily in someone that has that disease state. So that would be asymptomatic testing. So if you want to take a bunch of people in a clinical diagnostic test, those are people who said, I'm sick, I think I might have COVID, test me. In screening, we're looking at people and we're saying, do you have COVID? Let's find out, so we can understand a little bit more about individuals. And then surveillance is testing individuals to determine at the population level the incidence or prevalence of a disease. It's not necessarily focused on the direct benefit of that answer to the individual, because it's focused on the population, but individuals can gain some knowledge about their status. And this piece is not required to be conducted in a clinical lab. The FDA and the CDC and the White House have all said surveillance testing can happen in academic research labs. If you find someone that is positive, you need to refer them for a clinical diagnostic test. And so this was the niche that we wanted to fit into. And so the university, which there was a lot of uncertainty with the university on whether we could really do this. And there was a lot of convincing that had to be done and a lot of fact checking on what this regulatory space was. Once that was cleared, we started to really sprint. And Aaron was part of this process as well. So he knows how stressful this became. So really from July 15th to September 10th was our sprint to operationalize what we were doing. And it ain't easy, let me just say. I know ain't's not a real word, but I'm using it. So our research group, which consisted of all the people here, we took about 5 of those, and I want to highlight a couple. Both Laura Bix from the School of Packaging and Claudia Holtzman in epidemiology were not part of our bench group, but they were critical in making this program work. But everyone else on here has really worked crazy. I mean, Claudia Finkelstein, who's our director of health and wellness and a physician, was going out into cornfields collecting samples from people that they were meeting as part of our IRB protocol. So we've had people really burning the midnight oil in getting us this far. So those 5 people all moved on to this surveillance committee. And so you can see there's a lot of names here. These are people that are in healthcare, information technology, public health, epidemiology, information technology, both health and just standard data warehouse work. All students that were involved in this, and all together we had to figure out how to operationalize this process. And so the first thing we did was to operationalize the Saliva Kit. So we picked saliva as our sample of choice. So the first thing that we wanted to do was make sure it was easy to use, and that each component that was in this kit has functional utility. And you can see here laid out in this picture what the components of the kit are. There's a biohazard bag, a dropper, a small tube, and a large tube. The large spit tube is the one that that's the one you spit into. It's a wide mouth tube, so it's easy. The small tube, which I'll show you a little other picture of it, is where we collect the sample. The dropper is for you to transfer your spit from the large tube to the small tube, and then you place your small tube in the biohazard bag. And this box is actually part of a really critical point. We wanted to make sure that this system stabilized the sample. And we wanted to make sure it was inexpensive. This entire kit costs about $3. So when we started, we developed a prototype in collaboration with Laura Bix in the School of Packaging. This is a picture of these are their directions for the creation of the box. And the box was really important because not only does it have critical information and hold all of the material, but you can see these 2 starbursts on the right picture. Those hold the vials, the small and large vials, so that this box also becomes a stand. And that was really critical in making sure that we could take this product and make every single piece of it useful and ensure that we're going to have success when people use it. It's really hard to juggle a couple of tubes and a dropper, even if you're used to doing that in the lab. And so we worked with a company, Westrock, which Laura Bix had industry collaborations with, and they worked with us to get the branding onto the box. And we don't even have this box yet. We're going to have it next week, which we're excited to finally get it because we've been making a lot of other boxes. But you can see here, it still has the two starburst cuts in the top. It has all the MSU branding. It has the information on how to log your sample. And you can see right here how the box holds the large and small tubes and how that makes it really easy to use. The second company that we worked with was a company called Lowry Solutions. They were really, really critical in the early phase of developing our plan for logging samples and tracking them through the laboratory. They helped us to develop these stickers, and you can see the sticker has two sides to it. One is an auto ID spot, which is kind of like a QR code, and the other side has an alphanumeric 3-letter 2-number code, and it's connected by a small span of paper on the sticker. These are placed onto the small vials, and here is what it looks like once it's placed on the vial. And the reason that we have these two parts is the part that you can read is for our participants to log their sample, and the part that looks all weird and digital is the piece that we use to gun the sample into the laboratory. Also, some of the features of this small vial, you can see in here, this has the RNA shield, which is that preservative of the sample. So once your spit gets into here, the RNA shield stabilizes the sample. And then those 4 ceramic beads that you can see in there are important in the milling process of the sample once it comes into the laboratory. And we preload those into these vials to make it easy once they come into the lab to homogenize the sample. The next thing we had to do, so that was worked out, and that was a nightmare. The second nightmare was operationalizing sample pooling. So what is sample pooling? Let's talk about why you would want to pool samples together from individuals rather than just run a single sample. So a single, the pros of a single sample are you get an individual result. You don't need to take anything apart in order to determine if someone is positive or negative. It's very simple and straightforward. The cons are, if you're going to run 2,000 people a day, you have to run 2,000 individual samples. So that takes a lot of time, and it's very expensive. So the reagents for doing this kind of PCR work can run $10 to $15 per sample. And if you do 1,000 a day, that's $15,000 bucks. And we were trying to develop something to save the university money while still allowing them to do this kind of work. And there's a shortage of reagents and supplies, right? That we've heard all about national testing shortages, and we wanted to make sure we could minimize the use when these things are in high demand. A pooled sample, the pros are it minimizes time, it reduces reagents used, and it decreases our cost. The cons with a pooled sample is that you don't get an individual result without taking apart the pool. So if we get a pool that has 10 people in it and it shows up as positive, we have to then go and retest all 10 of those samples. So that takes time. And that might not be that big of a deal, but if incidence goes up, then the number of pools, the odds of any particular pool being positive are going to get higher and higher. And eventually you might have to take apart every pool if you get to a 10% positivity rate. And we have a loss of sensitivity, right? If one person's sample, only 1/10 of that sample belongs to a positive person, it's harder to detect that sample. So the question was, could we get an individual result from a pooled sample without taking that pool apart and rerunning every sample in it if it was positive? And you can do that. And we read about this. There were several papers that had come out about this. And this is an example from one of the TV segments that I did. And it was just easy to pull. So let me explain to you how transposition pooling works. If you've ever seen, and many of you may not, but the format that we use in the laboratory is a 96-well format. And so that the samples are generally in a plate, and they are 12 columns by 8 rows. And that's how this is set up. If you look here, it's an 8 by 12 matrix. And so think about the idea that you'd get one positive sample in this particular scenario. And that would be right here where these 2 red lines are intersecting. If you were to pool everything on this plate in one direction, so you take every one of these columns here and you put them all together into this one set right here, you pool them all together, and then you take all the rows and you pool all of them together based upon their columnar organization, you would end up with, in this case, 8 row pools and 12 column pools. And so you can turn this 96 wells into 20 different pools, right? So you have 12 here and 8 here. So that saves you 80%. And if you had one person positive, they're going to show up positive both in this pool and in this pool. And so we can trace those pools back to this location. And in that way, we just can do this mathematically and by coordinates without having to rerun either one of these pools. So everyone shows up in 2 pools. If you're positive in 2 pools, we know who you are. So this increases our throughput. We use 20% of the amplification reagents that we would normally use in a single sample, and each sample is unique to 2 pools, right? If 2 pools light up, we know who's responsible. Let's show you how this works. So let's say we have 768 samples. This is what we do in the lab. So this is a set of RNA from 96 people. We took their spit and we put them into, we took 96 people, we transferred them into a wash system, we created some RNA, and we put those 96 samples on this plate. And now we're gonna have a lot more than that because we have 768. So that's gonna be 8 plates like this RNA plate number 1. So this is how we do it. So the entire plate goes into one column. Then we get another plate, plate 2, that goes into the second column, plate 3, plate 4, plate 5, plate 6, plate 7, plate 8. So everything is moved into these column plates right here. And then we have a set of controls over in this part of the plate so that we can create positives and negatives and make sure that the plate is functioning appropriately. Then we do the same thing in the other direction. So we make a row plate with on row A, B, C, D, E, F, G, and H. And so each one of those plates, the column plate, which was that first one, and the row plate, which is the second one, they go into 2 separate PCR machines and we run them. And when that happens, you'll end up with 2 different locations. Let's say one person lights up. You're gonna see that particular person, we have this particular pool lights up and this particular pool lights up. Those are from, if you're in column 4, that's plate 4 right here. And you can see that would also be the 4th row down here. And then their position based upon how far they are down in the row or the column determines which individual they are. Here's another example. If this pool and this pool lights up, we know it belongs to this person on plate 7. And so this is what it looks like when you visualize these results. This would be what we would see on a typical amplification plate. We're going to see a lot of these sad humps, these flat lines that aren't really going anywhere. And then we're going to see some amplification. And so we're gonna take this and take it apart for you. So this would be a column plate and this would be a row plate from the same run. And here we're filtering out just an individual positive person from that. So here's the column plate positive pool, and this is the row plate positive pool. You go, okay, so what we're seeing here is 4 different rows, or 4 different amplification plots. We have an internal control, which is the yellow, and then 3 different genes that we're looking at for SARS-CoV-2, the S gene, the N gene, and the open reading frame 1 gene. And so here are those same column plates and row plates, and we have to talk about the issue between a column plate, and so we know who this person is, right? So the column plate shows us, with the row plate, we know who this person is. But we still want to examine each one of the positives that we find right now. We're still validating each one of those as a single. So we're not rerunning the entire pool, we're just taking the one that we think is positive and running it as a single and comparing it. And that's really important when it comes to sensitivity issues. So when I told you about the cycles that we use to amplify these samples, as you can see over on this one I'm circling here, it took 24 cycles for us to get this particular amplification on the column plate. This one has 12 samples in a column plate. The row plate, it only took us 22 cycles to amplify the material because there were less people in there. So the proportion from that one person, there's 8 people in this particular pool. So it's a slightly lower number. And then you can see here on the single sample by itself, we were able to amplify that at 20 cycles. So we don't really lose that much sensitivity when you think about this. The samples can be run out to 37 cycles. Once we get past 37 heating and cooling cycles, you start to amplify dust and you can't really use those samples reliably. The test that we use can go out to 37 cycles. And so it's pretty easy for us to see how someone who's positive at 20 alone would be positive at 22 or 24 cycles. It doesn't really take that many more cycles because it's an exponential growth phenomenon for us to be able to utilize this. So we don't lose too much sensitivity, and we were able to reinterrogate these samples because they're saliva. So what were our initial goals for the program? We wanted to operationalize an easy, non-invasive process to test for asymptomatic faculty, staff, and student volunteers on campus. Remember, this is not for symptomatic people, this is for asymptomatic people. We wanted to develop a system to collect, process, and analyze and transmit results on up to 10,000 samples a week. We did this in 8 weeks. We wanted to demonstrate academic labs can rapidly transition to assist in the fight against COVID-19. We wanted to instill confidence in MSU's ability to monitor students and staff and faculty and examine viral transmission. And we wanted to determine what groups and what behaviors are associated with asymptomatic spread. And we wanted to be able to take proactive steps to minimize spread on and around campus. Why examine asymptomatic spread if you're not going to make decisions to change the way that you are doing things? So I'm not going to go through this. This is just to show you how terribly complex this was when we started doing this. So we had various groups. We have the IT group, which deals with all of our data and logistics and messaging. We have the epidemiology team that Claudia Holtzman runs, which really deals with all the data issues with respect to behavior and demographics. Aaron Souza, who so graciously introduced me and is dean of the college, worked with me to coordinate and manage all the teams. We have a group that deals with transportation and logistics of getting samples into the lab and getting kits out of the lab. The laboratory itself that does its work, and then a communication strategy, because if we can't get people to volunteer for this program, there's no point in doing it. And I'm going to walk you through this entire process. So let's go through the movement on the system. What happens? So the first thing that happens is you get if you've registered for the program, you get a text that tells you it's time for you to get a kit and spit. And then it gives you information in your email. You go, you pick up one of these kits, which we've already shown you. You go through the sample acquisition process. So you spit into your tube, you move your sample into your little tube here, and then you take that code number that you can see on the file and you enter it twice in our system. You take that, you drop it in one of our drop boxes. Those samples get collected twice per day and are brought back to the bioengineering laboratory where we produced the saliva lab. The samples get gunned in. This is my favorite part. I really love this gun. So the fact that they can whip through these samples with those auto ID spots, they can log 96 samples in a minute, which makes it really easy to track these samples throughout the lab. And you can see in this is a 12x8 matrix. So just like I told you that 96 well plates, these are set up in the same configuration when they come in. These big vials need to be transferred into a much smaller format. This is someone taking the samples and moving them into the smaller format. They are washed and the RNA is extracted. The material is put in with the amplification materials into the reaction chambers. These plates are then scanned in and run in the system. And then the amplification data is examined. And then it is uploaded into the MSU electronic data warehouse. And the data is collected and aggregated. And then once that happens, a text goes back to the participant asking them to check their email so they can find out how they did on their saliva sample. Enrollment in the program right now is closer now to, I think, about 5,800 people. And the enrollment really grew once the president sent out a single subject email to implore individuals to sign up for the program. And it's really important to continue to effectively communicate. Remember, we only have 2,000 or 3,000 people on campus right now, living on campus, but we have plenty of people that are working on campus and students that are living all within 10 miles of campus. And so we're currently, if a student is willing to give us a sample, we will test them weekly. If they want to test less frequently, we'll do them every 2 weeks. Faculty and staff are every 2 weeks, but 3/4 of our samples are focused on student symptomatic or asymptomatic spread. And also here, the samples processed by day, you can see kind of our work week. This was in the very first week when we started, September 10th. We got a few samples, and then this is built up to anywhere from 600 to 1,100 samples per day at this point. So how did we do in meeting our original goals? Did we operationalize an easy non-invasive process to test asymptomatic faculty and staff and student volunteers? We sure did. Did we develop a system to collect, process, and analyze and transmit results for up to 10,000 samples per week? Yes, we did. We demonstrated that academic labs can rapidly transition to assist in the fight against COVID-19 and operationalize a system like this. I think we've instilled confidence in MSU's ability to monitor staff and students and faculty virus transmission. Did we determine which groups and what behaviors are associated with asymptomatic SARS-CoV-2 transmission? Not yet. We actually don't have access to this data yet. And I think this is one of the things that is unforeseen is once we collect all of this data, who has access to that data, who should be allowed to see it? So that hasn't happened yet. And we're in the process of dealing with that. Maybe in the next week or so, we're going to have decisions on this, but we didn't really think about how complex of a situation this is with respect to people's privacy and what information we should be sharing with the public versus information that is needed for operational management of the university. Take proactive steps to minimize spread on and around campus. It's hard to do that if we don't have access to that data yet. So the epidemiologic team needs to get access to that data, and they can then offer up those those pieces once they're analyzed to leadership. So that's still waiting to happen. Can we detect outbreaks sooner? I think that was the title of this entire talk. Maybe, I don't know. But based upon our anecdotal data on some testing we've done for athletics and from self-reporting of individuals that are volunteering to tell us if they have a positive sample from us and whether they end up subsequently positive with an NP swab, it appears that we are able to catch people in the very early phases of the disease. So what are the remaining challenges that we have? Who gets access to the testing and behavioral data we've generated, right? That's the big question right now. How will these data be paired with wastewater testing for surveillance? The other piece of the work that's going on at MSU is wastewater testing with Joan Rose and Jade Mitchell, and exactly how that data is going to be paired with our data to see if the data that we're demonstrating with saliva can be predicted by wastewater monitoring on campus is yet to be done. How will MSU utilize that data to inform decision-making? How will they turn this data into action? We still don't know yet. How to best determine the utilization of this asset and where it's needed most, right? We've developed a system, we are using it on campus, it is valuable for us. What can we do with it beyond MSU? What about bringing back experiential learning for students that are in professional schools like the medical school or the vet school? What about outlying campuses, places like Grand Rapids? We'd originally developed this program for a Grand Rapids return to work process, and it's only in the East Lansing right now. So can we move this to outlying campuses? And what about communities beyond MSU? What about schools? What about the under and uninsured? What can we do to help them? And then how do the rest of us get back to our day jobs, because we have other things we have to do. So turning this into a self-sustaining process that can be run by others by making the operations that we've produced robust and reproducible for other people to run. And so beyond those early goals, we've learned a couple of other things, that we have a rapidly deployable system that was developed with university intellectual and technological assets that were developed in collaboration with industry specialists and domain experts all around campus. We developed a cost-effective novel saliva kit for $3, a low per sample run cost for the direct materials and labor for the amplification and detection of the virus is about $8, and the potential for scalability into a statewide network of labs positioned to conduct surveillance testing is a real possibility, right? We could take this and work with others to develop a similar system and have centralized reporting to develop a real web of monitoring sites all around the state. And we think it's a really robust model, and it's working well, and we'd love to share it. And then finally, is there any value, even though there's no real intellectual property in this, the actual production and distribution of the spit kit, which is $3. Rutgers developed its own kit with a company called Spectrum LLC, which was a venture capital. Rutgers is selling $200 million per month of their saliva kit. So there's some upside potential of this if we want to scale it more widely. I cannot say I did all of this alone, because I absolutely did not. I was just the stubborn person that started it, along with Joe. So right here, Joe in his mask, he's really the smartest mouthiest of us all. And he's the one who got us all into trouble doing this process. All the rest of these people are people from the surveillance team. And of course, right down the center, you can see this is the new laboratory in the bioengineering building, which takes up half the floor in the IQ. And these are new individuals that are working specifically on this program, constructing saliva kits and running the samples every day. And then of course, here's Aaron, because I had to beg Aaron to get his picture I wanted everyone to give me a selfie, but he was the last person, so now he's a cool kid with the rest of us. So thank you. Make sure you give a spit. If you are a member of the MSU community, go to go.msu.edu/SpartanSpit to learn more. And if you want to enroll, go.msu.edu/Spartan-Spit-register. Thanks.
Thanks, Jack. That was excellent. We do have a couple quick questions. The first one I believe we've already answered, and that is this only available in the Greater Lansing area?
Yes, for now, that's it. I think the issue is how to operationalize it to outlying communities. right now we collect samples 5 days per week, and getting them from wherever they are to us, I think is a large logistical challenge, particularly if the volume of samples aren't high. I think that the Grand Rapids community is really going to be the next potential beneficiary of this. But then we have students all over the state for CHM, and the farther out and the fewer there are, the more difficult it is to operationalize it, which is why I'd love to have a network of labs doing this around the state.
A very similar question. Because you are using an RNA shield, do samples need to be returned the same day you supply the sample? For example, spit Sunday and return the sample Monday?
No, the RNA shield is great. So you put it into that vial and you shake it vigorously for 15 seconds, and that sample is good for weeks. We've tested and retested samples that have been sitting on a shelf for 6 weeks, and there's no degradation of the sample. We've put these at 100 degrees and let them sit for a week, and there's no degradation of sample. The only time you really get a degradation is from freeze thaw. So if you take these and put them in the freezer once, they're okay. If you put them in twice, they start to degrade. So that's really the main challenge. No, they're really stable.
Great. How has communication and raising awareness with students went?
Well, this is really a great question, and it's hard to answer right now. So students have enrolled. Students are the majority of our participants. The students that are involved, I help man the shared mailbox for everything that goes wrong with this program, spartanspit@msu.edu. And so every challenge I see, and I've answered a good portion of the questions that come in on that line. The students are concerned about getting their tests back, but then when you answer them and give them some information, they are the most grateful and excited people I've seen. Whether or not the students that we have that are enrolled in this program are typical of the average MSU student, I don't know. They are self-selecting to be part of the program. As volunteers, they are motivated to do this. And whether they are of the same behavioral profile as students that we are seeing a high prevalence of SARS-CoV-2 transmission in, we don't necessarily know. The level of positivity that we see among asymptomatic volunteers in our program is much lower than what MSU is seeing or what Ingham County is seeing. So I'm guessing we have a lot more well-behaved people in the program. But the analysis of that population is really going to be key to understand whether continuing as a voluntary program is worthwhile, or whether the university needs to re-examine the concept of making this more mandatory.
Excellent. Sorry if this is already addressed, but if you do get a positive sample, do you recommend the individual follow up with an accredited lab to confirm? And do you provide any guidance such as quarantine, telling contacts, any of those?
So the information that we provide is very, very specific. When we give information to a participant, because we are surveillance, we cannot give them a diagnosis. So we can only tell them, based upon our examination of your sample, you need to seek a follow-up clinical diagnostic test. And we provide them with connections to the MSU COVID-19 hotline, to a triage hotline, to all of the links for local tests. If they're students, we connect them to Olin or to the stadium where samples are collected for Sparrow, and we give them all the information. The only thing is that there's no contact tracing, and there's no informing of the health department from our work, because our work is in clinical diagnostic. We don't provide it as part of the public health apparatus. So there's no contact tracing that's going on from this. But if people are doing their job and they are following our directives, they are then going to get a test, they will get confirmed, and then they will fall into the contact tracing and public health apparatus that exists.
Would they then be taken out of your group once they're positive?
Once they are positive, we ask them to no longer participate in the program, and the system will boot them once they are positive. Because we want to be able to look at prevalence in people that have not had the disease. And if we keep people in that are already positive or have been previously positive, then we are probably artificially lowering our perceived prevalence rate, because those people are never going to be positive for us again. At least we don't think so.
Great. What is your ultimate goal? Would it be testing at the state level, national level?
My ultimate goal is to develop a giant network of academic labs that can respond to crises just like this. And I've put this out a couple of times, kind of the development at the federal level of a biomedical National Guard that can be activated quickly in situations like this. If laboratories have the technical know-how and they have the equipment, and we can develop an organized response to this particular pandemic and to other biological crises, we can respond so much quicker. And we don't need to have the Army Corps of Engineers figure out how to do this, or have Quest Diagnostics spend $1 billion to grow its footprint, when we have all of these laboratories that are available that could do this at 200 a day, 500 a day, 1,000 a day. If we can develop a plan and a process that's easy for people to follow, then they have a roadmap to transform their work into monitoring as opposed to research.
Great. This next question comes from an attendee who's writing a story on this webinar. So that's great. Thank you. If you have one hope from today's presentation, what is it?
Oh, that people didn't fall asleep mainly. But if I had one hope from what I'm hoping to get out of what we're doing, it is to raise awareness of the awesome talent that exists at this university to be able to put something like this together, and to remember that we are not just here to come up with answers in theory or hypothesize on things that could be done. We need to be able to demonstrate that we can implement things. And I'm in a department called Translational Neuroscience. Translation is the bench to bedside concept. And so if we couldn't take this and operationalize it, I have no business being in a translational department. So translation is key, and being able to utilize the assets of a university to create community good. That's really what I want to be able to do. And I think that's what we've shown we can do.
Love that. What happens if multiple samples on one plate test positive? Can you narrow this down to identify the multiple individuals that may be COVID positive?
Yeah, so it's really interesting. We've had more than one person positive in a particular pool. So think about if you have 2 positive samples in a pool, that pool just lights up once. But that pool has two sister pools, one for one person who is in that pool, and one that intersects for another person. So if you have 2 people in one pool, you're going to get 3 pools lighting up. We can take it apart unless it gets really, really messy and we have a lot of people. Then it becomes difficult for us to take apart a pool. But with positivity rates under 1% in the asymptomatic, it has not been difficult. One of the problems that we have had, though, is when samples come in, and we didn't really even think about this, we have 10 or 11 locations where we're pulling in samples from. The samples that are positive tend to come from one place. And so if we open them up, the samples come in and we rack them based upon them coming in from a similar location, we end up with a bunch of samples that can come in together that are the same. And so we found that we had to take the samples from everywhere, mix them all together, like in one of those bingo wheels, drums, kind of mix them all up and then redistribute them so that they can be spread out appropriately. And even though we have that problem on occasion, remember, we still are going through and saying anything that we have as a hypothesized positive. So anyone who would show up in 2 pools, even if they're there as a bystander because of 2 other people, we run all of their samples. We have that saliva sample. We can go back and reinterrogate it. We run them all as singles at the end of the day, and we confirm them. So every sample gets run 3 times, twice in a pool, once as an individual if they're positive.
And I think you kind of touched on this, but how prevalent would COVID have to get to before you're no longer able to really make this work?
So for pooling, you can still make pooling work unless it gets really crazy. You just have to shrink the size of the pools. So if we get over about a 5% prevalence rate, we would move from pools of 12 and 8 to pools of 6 and 4. We would end up using 40% of the materials instead of 20% of the materials. But we would still be able to then, by having smaller pools, the odds of having more than one sample in a pool go down significantly.
Great, and it looks like we have one final question. Do you guys work with any clinical labs in the Lansing area or the health department lab?
The only laboratory that we are currently working with is Olin. So John Gerlach is the director of the Olin lab, and he oversees the vet diagnostic lab as well, because he has a CLIA license for Olin to run these tests. And he is currently working with us to validate our tests clinically. So the saliva test and the way that we are doing the Thermo Fisher test that we use for the 3 genes that I laid out, the N, the S, and the open reading frame gene, those together need to be validated. And once we get those validated, the Olin lab is looking at extending its CLIA license over us. And so that those confirmation samples that we run, the second set of single samples, we can run those under a clinical diagnostic framework, and then give people an actual diagnosis at the end of this, and then move our program from surveillance into screening.
Great, thank you. That is all the questions that we have for today, and we are just a couple minutes till 3 o'clock, so I think we can wrap up and let you get back to your great work. Thank you for everything. It was an excellent talk, and we appreciate you.
Hey, thank you very much for having me. I appreciate everyone spending an hour listening to me blather.
Oh, hold on one second. We have one question just came in. Is it worth it for GR faculty to register?
I think that right now, if the inclusion criteria are having to come to campus on East Lansing once per week. If you go to East Lansing once per week, you can be in there even if you're Grand Rapids faculty. I think once we are ready, and this is something that we're trying to operationalize with Aaron, to develop an opportunity for us to do samples in Grand Rapids, then once we have that going, we will announce it. We'll make sure that people know they can register and become part of the program.
Sounds good. Thank you.
All right. Thanks very much.